the mRNA processes differ from a eukaryotic cell to a prokaryotic cell. We're going to be discussing the characteristics of eukaryotic mRNA right now. Starting off, we need to talk about a term that is primary transcript, which is the RNA directly transcribed. So this is transcription we're talking about. So during transcription, a template strand of DNA was read, transcribed to form a strand of RNA. This strand of RNA that is immediately produced during transcription is the primary transcript. It has not been modified further. It's just the RNA produced from it. Now, why do we make the distinction from the primary transcript to the mRNA? And that is because a primary transcript is not always mRNA. Yeah, there are different kinds of RNA. There is ribosomal RNA, there's transfer RNA, there's something called small nuclear RNA, and there are other RNAs as well. So our primary transcript, our RNA that's directly produced from transcription, is modified according to the role that it should be. So it is modified to form that small nuclear RNA, to form that rRNA, to form that tRNA. And right now we're going to be talking about a little bit about the general processes by which our primary transcript is modified to produce mRNA. Messenger RNA, which is the RNA used for protein. So our mRNA, for eukaryotes, we will see coming up that for prokaryotes is different, but our mRNA is not equal to our primary transcript because our primary transcript was modified in order to make the mRNA. They are now not equal to each other. Now, what are these modifications that we make? There are 3 of them that you should be aware about. So let's draw a quick model. Let's say that this is our primary transcript that is destined to become our mRNA. We need to make a couple of modifications to actually make it mRNA. So we'll start off with the 5' cap. A protein will come in, it will add a molecule called methylguanosine to the 5' end. You don't need to know the specific name of it, just like maybe be aware that something is added to the 5' end. And this thing serves a couple of different purposes. I'll talk about 2 of them a little bit. One is that it offers further stability to the RNA. And also, the presence of it allows it to interact with new molecules compared to if it wasn't on there. And we also add something to the 3' end. We add a 3' polyadenosine tail. You don't need to really know the poly A, but the reason why it's called poly A, where poly means multiple, that's because a bunch of adenosines, the nucleotide or the base, or nucleotides carrying the base, which is the adenosine, are added to the end of the 3' end. And these serve the same purpose, which would be stability, interaction with molecules, and whatnot. So just be aware of these. You don't need to know specifically what is added, but whatnot. The 3rd one is the one that you should be most aware about, which is alternative splicing. In eukaryotes, not everything of a primary transcript that is meant for proteins is translated, meaning before our mRNA is read by a ribosome to produce the amino acid chain to make the protein, it is cut up to remove parts that are not wanted. So it's cut up, parts of the RNA are removed, and only the RNA left over is actually translated into protein. So it's kind of like we're removing the potential of the amino acids that the parts that we moved coded for. And we'll talk about this in a little more detail in just a second. But to summarize, we had our DNA, right? Our promoter indicated what section of the DNA that we should transcribe. And so in the process of transcription, we made RNA. But this is our primary transcript. And we're talking about the path to produce a protein. So this is an mRNA we're talking about. So we do additional modification. This additional modification with these three things. And then after that, we get our mRNA. And then this mRNA is translated into protein. So this means that our mRNA was used to make the protein, not our primary transcript, because it was modified. So this is for eukaryotes. Let's talk more about alternative splicing. Eukaryotic cells are able to modify their RNA, their primary transcript, to such that different mRNAs can be yielded. This means that eukaryotic cells can remove the RNA that is not wanted to be translated and only leave the RNA that is wanted to be translated. So this RNA that is wanted and that is kept are called exons. And the RNA segments and stretches that are not wanted are called introns. So to write down, exons are the RNA kept in mRNA, and the introns are the RNA removed, not seen in mRNA. Okay, so this is a model of a RNA that was transcribed, and it is not yet that mRNA. We added that 5' cap and we added the 3' tail, but we didn't do the splicing yet. Let's say that this is our intron, and that this is our exon, and that this is another exon. Okay, what does this mean? This means that the intron is what we want to remove, and the exons are what we want to keep. So what will happen is a protein will come and it will sever the phosphodiester bond over here. It will sever the phosphodiester bond over here, and then we can model this a little bit. And after we do that, it can be removed. So now that it is removed, there is a gap, and then what happens is another protein will come along, and it'll make a new phosphodiester bond between the existing exons, and it will splice them together, remake that phosphodiester bond. And let's say that that was our only intron, so now we're done. We did all the steps necessary to convert our primary transcript to our mRNA, the 5' cap, the 3' tail, we removed the intron. So our ribosome will come along, it'll read the mRNA, and then it will translate the codons into amino acids. This means that the exons that were kept, the DNA that is kept, are used to actually make the different amino acids in the correct order. This means that our exons are the portion of RNA that are expressed, where expression is determined by it being translated. So our intron was not expressed. It was removed before it could be translated. Now, what do I mean by alternative splicing? Let's say that, okay, let me do a couple minutes. Let's say that this was not the intron, that this was an exon, and that and that this entire thing is an intron. So we did the exact same thing that we did earlier. The protein will come, it'll cut on both sides, and then it'll splice together the remaining exons like that. So you see that this is a truncated mRNA, which will result in a different protein. But in both instances, the RNA was spliced to form the mRNA, but they were spliced in different ways. So RNA can be spliced in alternative ways, like not just one way always. and this will produce a different mRNA. And because there's a different mRNA, it will produce a different protein. So alternative splicing, that means that eukaryotes are able to remove what they don't want, keep what they want, and they can do these in like different ways. A way to remember exons from introns is that exons are expressed. They're the ones that are actually translated. And introns are not expressed, they're removed. The processes of prokaryotic mRNA differ in a number of ways compared to those of eukaryotic mRNA. So to start it off, there is no additional RNA processing needed to go from the primary transcript to mRNA. So whenever in eukaryotic cells, you need to put on the 5' cap, you need to put on the 3' cap, you need to cut it up a bunch, and that's splicing. You don't do any of that. If the primary transcript is meant to form the mRNA, it'll just be the mRNA. Now, the primary transcript is not always mRNA. You know, prokaryotes still need to make rRNA and tRNA. But if the primary transcript was meant for mRNA, you don't need to do anything. It's just mRNA. So another thing is that prokaryotic mRNA is polycistronic. This means that a given mRNA will produce mRNA encodes multiple genes / proteins. Generally, the mRNA produced from eukaryotes are monocistronic, meaning they only encode one gene, and that means that they only produce one protein from the mRNA. But a lot of prokaryotic mRNA are polycistronic, meaning that multiple proteins are produced from a single mRNA. And the third thing is that transcription is coupled with translation. This means that as the RNA polymerase is transcribing mRNA from the DNA, ribosomes are attached to that mRNA and they're translating as it goes. So a question on the exam might ask you, okay, given this model, what are we looking at? And the model for prokaryotic mRNA probably looks something like this. You would have your DNA, and then your RNA would be transcribed at multiple locations because it is polycistronic. And then because they're coupled, you would probably see like little ribosomes hanging off, and they're also producing their own thing. So this would be a crude model of prokaryotic mRNA. Okay, let's briefly cover the differences between the mRNA processes for eukaryotic cells and prokaryotic cells. So for eukaryotic cells, the primary transcript is not equal to the mRNA. So there is additional modification needed. Where for prokaryotes, the primary transcript, if it is meant to be mRNA, is just mRNA. So no additional work. Okay, eukaryotic mRNA is generally monocistronic. This means per mRNA, one gene, meaning one protein, or one kind of protein is being made. Prokaryotic mRNA a lot of times is polycistronic, poly meaning multiple. So per mRNA, multiple genes, which means multiple gene products, which would be the multiple proteins. I'll say multiple kinds of proteins. Eukaryotic cells generally have separate transcription transcription and translation, where prokaryotes have coupled. Yeah, transcription and translation, meaning that as the DNA is being transcribed, or as the RNA is being transcribed, it is also being translated.